second, going into presenter mode. And now you can also see my screen and the laser pointer, right?
Yep.
Okay, so thank you very much again, Neva. And it's a pleasure to be with you online. So until quite recently, until just a few years ago, protein design was considered to be mostly a basic research question with very few or almost non-existent practical applications. But over the past few years, we've seen that our methods have become quite reliable, and that has enabled us to offer real solutions to challenging problems in protein engineering. So during the first part of my talk, I'll describe how improved understanding of how proteins fold and function has led to the development of very reliable tools for improving protein activities. And in the second part of my talk, I'll discuss still unpublished methods that we've developed in order to design massive repertoires comprising millions of designed proteins, and specifically enzyme repertoires. And I'll show how these repertoires are surprisingly effective in sort of exploring the functional space that's open to various proteins. So before I start, we should all acknowledge that proteins are already very critical for modern research and industry, all the way from, let's say, paper making to the making of life-saving drugs. Enzymes and other proteins have a major contribution. And it's very clear that the chemical industry will have to shift increasingly to using enzymes if it is to become more green, energy efficient, and also more sustainable. So proteins are clearly great and very useful, but as a very general rule, no natural protein is ever good enough to serve in real-world applications. And this may be because of various reasons, starting from the thermal stability, meaning proteins are only marginally stable. I'll talk about that. Their recombinant expression levels, our ability to produce them in common lab hosts is low. And also their activities may be limited, activity specificity, and also stability in various reaction conditions. All of these are very serious concerns when we want to apply a protein either in research or in a real world, let's say, industrial application. The underlying reason for this is that proteins are large and complex. In fact, they are the most complex molecules we know. They're also the most beautiful and useful, so we still need to take care of them. So how do you deal with such complex molecules? How do you optimize them? So today, the main strategy for optimizing protein activity or stability relies on Darwinian optimization or evolutionary strategies. You start with some low level of activity or stability of typically a natural protein. You introduce diversity, let's say by mutagenesis, and then you screen for improved activity or stability. Now, because a single mutation never really reaches the levels that you'd like to be at, you have to iterate this process. You find the best a variant from this library, and you introduce diversity again and again and again until you reach a peak. Now, you clearly recognize that this is a greedy algorithm. It follows the path of greatest improvement. And like all greedy algorithms, it can reach a plateau very, very quickly. If that happens, of course, you have to rewind the process and restart in the hopes of getting to a better peak. So evolutionary processes are great. We're all here thanks to evolutionary processes, but they're also very time consuming. They rely on chance and can become stuck in a local optimum, as you see here. They also, in order to implement them in the lab, they demand expertise, and often they require rounds and rounds of iterations through thousands, if not tens of thousands and millions of different variants. But really, most critically for us as scientists is that relying on chance, as we do in these processes, admits critical gaps in our understanding of how function is encoded in proteins. And this literally gets us to the heart of the matter, which was phrased incredibly beautifully by Richard Feynman: what I cannot create, I do not understand. The fact that we still use chance in order to optimize, let's say, life-saving drugs such as antibodies means that we don't really understand how proteins fold and function. And perhaps the defining question for protein design, if we understand this, is that we need to develop really a general and reliable strategy to control and optimize protein activity. And this is sort of the goal that we set ourselves. In this slide, I'm trying to summarize really all of our work in the lab for the past 10 years and beyond. So like many other labs, we use atomistic design calculations using Rosetta. In these design calculations, we can generate many different models of structures, and we analyze each one in terms of physics-based energetics, such as van der Waals, hydrogen bonding, and electrostatics. But as many have observed, including ourselves, such atomistic design calculations on their own are not reliable enough to design large and complex proteins, such as antibodies, enzymes, and other proteins that we're interested in. For that reason, we developed the unique strategy that we're developing, which we call evolution-guided atomistic design, also uses information in public databases, such as sequence homologs of the target protein, let's say all of the homologs of a specific enzyme, and experimentally determined backbone conformations in order to guide the atomistic design strategy. So this is sort of a hybrid evolution-guided atomistic design strategy. And increasingly, and as I'll describe in my talk, we're also using machine learning and AI in order to integrate large amounts of computations and experimental data in order to design very large and effective repertoires of designed proteins. So that's very basically a schematic of how we do things. So maybe the most fundamental and important problem in protein design is the question of protein stability. Many natural proteins are only marginally stable, which means that many of them cannot even be expressed to high quantities. So for instance, from the structural genomics initiatives, we know that in a typical proteome, more than 50% of non-member proteins are insoluble. Even though they are cytosolic proteins, they're very difficult to overexpress in expression hosts. We need to find a way to stabilize such proteins, and very critically, without reducing their activity, because a reduction in activity typically makes that protein completely useless. So we must find a way to stabilize the protein on the one hand without impacting its activity. And again, a very critical point is that we want general rules that can be applied universally to stabilize proteins. We're not interested in a method that could be applied to only one type of protein. Ideally, we'd like to be able to work on all types of proteins, be they cytoplasmic, membrane proteins, enzymes, antibodies, what have you. So to that end, we developed a method called PROS, for protein repair one-stop shop. And before I describe results from this method, if we want to understand, if we want to develop a general method, we need to understand why so many proteins are only marginally stable. The reason for that can be seen in schematics such as this, which are called the energy landscape for the folding of a protein. Now we know that the native state, which is also the active state of a protein, is always going to be lower in energy than any of the alternative misfolded or unfolded states of a protein, even in a marginally stable protein. That's going to be true for such a protein as well. But for a large and complex protein, the number of misfolded states may be huge, and the protein may be trapped in any one of them as it tries to fold into the native state. Now, our design goal, if we want to stabilize the protein, is that we want to accentuate the gap between the native state and any of the misfolded and unfolded states, and to eliminate as many of the misfolded states as possible, so that we're left only, if possible, only with the native state. Now, what is the problem with such a goal? Obviously, we have information, atomic information about the native state. We have, let's say, the crystal structure, or now an AlphaFold model of the native state, so that's fine. But we have absolutely no information about any of these misfolded states. And again, these may be numerous. And again, we don't know anything about them. So how can you use an atomistic design strategy in order to counter these misfolded states? This is not possible. So in this case, what we have done is to use evolutionary data in order to counter such undesirable misfolded states. The logic for this is as follows. So we know that for any native protein, any naturally occurring protein, that protein has evolved in very different contexts, let's say from fern to frog and other organisms. And we know that in all of these organisms, the homologs of that protein must have maintained the ability to fold into the native state and to carry out the primary activity of that protein. Of course, there are differences in selectivity and stability or what have you, but the primary activity of the protein and its ability to fold into the native state must have been conserved through evolution. And that means that we can use evolutionary data in order to guide the atomistic design calculations and stay away from mutations that might improve or really cause the protein to misfold or not fold correctly into its native state. And this is the idea behind PROS. So without going into the details of how the method works, all of the papers will be mentioned in this talk, in the presentation. The first example in which we use PROS or tested it is human acetylcholine esterase. The enzyme structure is shown here. This is an essential and very challenging enzyme to work with. It has an essential role in neuromuscular junctions. It's also the target of nerve agents and also other drugs. And for decades, people had tried to increase or find a variant of acetylcholine esterase that could be expressed in common microbial hosts to use it as, for instance, for drug discovery. But this protein is also very challenging to work with. It's more than 500 amino acids in length. It has multiple disulfide bonds shown here and here. It also has multiple glycosylation sites, and its active site, shown here in yellow, is buried some 20 angstroms below the surface of the enzyme, meaning that the activity of the enzyme is very sensitive to mutations anywhere in the enzyme. It's a very sensitive enzyme to mutation. So we knew that this is a very challenging case, and yet we applied PROS to it as the first example. We tested just five different constructs. Each of the constructs that PROS designs encodes typically dozens of mutations relative to the wild type protein. In this case, the best design incorporated 51 simultaneous mutations relative to the wild type or human enzyme. And as you can see here in these orange spheres, they're encoded throughout the enzyme domain, of course, outside of the catalytic pocket. We don't want to interfere or impact on activity, so we only design other parts of the enzyme. But we do design the core of the enzyme, for instance, space-filling mutations such as this. The mutations are shown in green. The wild type is in purple. You can see that some of the mutations fill spaces within the core of the protein. Some of them rigidify the loop or introduce new bonds, et cetera, et cetera. These are what you would expect stabilizing mutations to do. They increase the number of contacts, advantageous contacts within the native state of the protein. But before our work, there was no other description of a method that could introduce so many simultaneous mutations to a protein without failing. At most, people tried about 5, let's say, mutations. Even in those cases, most of these designs would fail. So here we introduced 51 mutations. What we saw immediately is that all of the mutants we tested, and again, these were just 5 constructs, exhibited about 20 degrees higher thermal stability than the wild type enzyme. The wild type enzyme is stable to about 42 degrees Celsius, whereas each of the designs we tested is stable to about 60 to 65 degrees Celsius. So this was very encouraging. Also, all of these designs maintained the activity. They didn't show any change in activity. They were still diffusion limited acetylcholine esterase, despite dozens of mutations from the wild type. But really, the most dramatic impact of these mutations were on the bacterial expression levels of these designed enzymes. Here you can see the full change of each of these designs relative to human acetylcholine esterase. So human acetylcholine esterase, as I mentioned, is very difficult, almost impossible to express in bacterial hosts. But you can see that each of these 5 designs expresses to at least 100-fold higher levels than the human. And this design, which is the same one I showed earlier with 51 mutations, expresses to almost 2,000-fold higher levels in standard bacterial cells than the human. So these were unprecedented results for a completely automated design method, and they were very encouraging. Since then, we implemented this method as a web server, and we've had literally thousands of different users, and there are dozens of papers and patents reporting the use of this method, successful use of this method. So it's been very useful. But this approach requires an atomistic and quite accurate molecular structure. And that in itself presents a problem because many times proteins are so unstable that they cannot be expressed to large amounts to enable crystallography. But 2 years ago, we were all awed by the publication of these AI-based methods for structure prediction, including AlphaFold. This is literally a watershed for molecular biology. This has been described many, many times. I won't go into the details, but we asked, so these methods are very accurate. They're about at the accuracy level of a crystallographic structure of about 4 angstrom resolution. So they're not perfect, but they are absolutely a major improvement on what we had before. So the question was, are they accurate enough to serve as starting points for design? Keeping in mind that the sort of mutations that we introduce are also in the core of the protein, where we introduce new networks of polar interactions or hydrophobic interactions, and such interactions may be very, very sensitive to minute details in the structure. So if the model structure is even slightly wrong, we may be in trouble using these methods. So here we targeted family of enzyme known as versatile peroxidases. These are fungal enzymes that can degrade lignin and attract a lot of attention for their ability to decompose lignin and release sugars and other valuable chemicals from lignin. But they're also incredibly difficult to work with, and to date, only one VP has been characterized in the lab and analyzed crystallographically. So a very challenging but important family of enzymes. We selected 10 different VPs using standard phylogenetic clustering. We then modeled them using one of these AI-based methods as you see here. This is a good point to introduce the fact that these enzymes also depend on heme and a variety of different metals. So this is a cause for concern because these methods do not model such cofactors, metals, heme, etc. And yet the models seem to us incredibly accurate, even in the heme binding pocket and around the metal binding sites. So this was very encouraging. Then we literally just took these models and for each of them designed 3 PROS variants. And as usual, PROS introduces dozens of mutations, and we tested them experimentally. What you can see here are 4 examples of VPs that can hardly be expressed in yeast. You see the wild type can hardly be expressed in yeast. This is in the gray bars. Yet the designs are massively expressed in yeast, even though, and these are the highest mutational loads. These variants typically encode 40 plus. 40, more than 40 mutations relative to the wild type and are based on model structures, and yet they express much better than the wild type enzymes. And for the first time, they allowed us to characterize these enzymes' activities, and we showed that these enzymes have very different selectivity profiles. I won't go into the details, but these selectivity profiles are very interesting on their own. But the take-home message from this is that now, for the first time, atomistic design calculations can be applied to genomic databases at very large scale. So as you know, AlphaFold has been used to compute the structures of more than 100 million different proteins for which we only have sequence information. Now you can download these model structures and directly use them as starting points in our PROS web server. And what we've seen since publishing this paper is that literally more than 1/3 of the users now actually use AlphaFold models on our servers. So this is very exciting. I think this will solve many bottlenecks in protein engineering and design. But PROS only stabilizes the protein, of course, as I mentioned earlier, by introducing mutations outside of the active site pocket. In many cases of protein engineering, we're actually interested in enhancing the activity of the enzyme or the binding protein. In such a case, we cannot just mutate the surface or of the protein or introduce mutations outside of the active site. We actually have to go into the active site and introduce mutations in the most sensitive part of the protein. Why is this challenging? So this is an example of an active site of an enzyme that breaks down xylan. It's called xylanase. I'll come back to xylanases in the next part of my talk as well. But essentially, any enzyme we look at shows the same, and also binding proteins shows the same properties. You see that the active site residues, shown here in blue, form very dense interactions with the ligand, which is shown here in green. But in addition to these critical interactions with the ligand, these amino acid positions also form very dense interaction networks among themselves. And you can imagine that mutating even one position here, just introducing a single point mutation, can cause the complete collapse of this constellation of amino acid residues and lead to orders of magnitude decrease in catalytic efficiency or binding affinity if this were a binder. So clearly this is going to be a very difficult optimization problem, but evolution does address this problem. It does optimize enzymes. How does it do it? Exactly in the same way I mentioned earlier, one step at a time. You introduce diversity and screen for the best variant, et cetera, et cetera, et cetera. But you can imagine, whereas this strategy relies on the introduction of a single mutation and verifying that that mutation improves the activity, and then introducing on top of that another mutation, you can see by looking at this very dense constellation of amino acid residues that this strategy, this greedy strategy, may fail because we may have to introduce doubles, triples, or even quadruples of mutations in order to reach an activity peak, whereas each of the single point mutations may actually be deleterious. So the question is, can we go beyond what evolution enables, these single mutation trajectories, and introduce, let's say, triples and quadruples of mutations that improve activity? For that, of course, the computational design methods need to be much more reliable than they have been in the past. So I'll describe how FuncLib works. Our model system for this was an enzyme, a natural enzyme called phosphotriesterase, or PTE. This enzyme breaks down this pesticide called para-oxan very, very efficiently, but it's also a very broad spectrum and promiscuous hydrolase. It also breaks down various lactones and esters, as you see here. What's interesting for us, or for human use, is that PTE also breaks down organophosphorus nerve agents, such as VX, serine, soman, and other very toxic compounds. How toxic are these compounds? They're actually the most toxic chemicals we know, and one drop on the skin of an adult can kill within minutes. So there's a lot of interest in developing an effective countermeasure against these very toxic compounds, but PTE's activity against these compounds is very, very low. It's orders of magnitude lower than what you would need for effective decontamination of organophosphorus nerve agents. So we asked the question, can we design PTE, improve PTE's activity relative to these very toxic nerve agents? Now, before I show you how FuncLib works, I'd like you to appreciate that these compounds are very different from one another, and also that they have many rotatable bonds, which means that we don't actually know how these compounds dock within the structure of the enzyme. We don't actually know how they would go inside the structure of the enzyme, and therefore we have limited understanding of how to design an enzyme against this. So here we ask the question, which is a little bit strange for enzyme design, can we optimize function without targeting a specific substrate? Since we don't know exactly how the substrates dock inside the pocket, we ask the question, can we design various variants of this of PTE without targeting any specific substrate? And in order to do that, in order to develop a general strategy for designing improved enzymes against an unknown substrate, we had to go back to the basics of what makes a very efficient enzyme. And what we know is that all high efficiency enzymes, regardless of the class of enzyme, we know that one, they have to be stable, because if they're not stable, they're not going to be active. And two, the core catalytic residues, those residues that are specifically responsible for the catalytic step, those must be pre-organized for activity. Of course, not all enzymes are pre-organized for activity, but those enzymes would typically not be high efficiency. High efficiency enzymes have pre-organized cores for activity. So the key question we ask here is, can we design enzymes that are stable? Well, that's easy for us to do now because we have PROS. We just use PROS to design a more stable PTE variant. But can we design catalytic cores that are pre-organized for activity? And the FuncLib solution is as follows. We fix those amino acids that are directly responsible for the catalytic step. Those are shown here in gray. We fix those exactly as we observe them in the crystallographic structure. We don't mutate them, of course, but we also don't even change their conformation 1 angstrom. We leave them exactly as we observe them in the crystal structure. By contrast, all the other amino acid positions that face the cavity of the enzyme, we allow those to mutate, and we generate many, many different constellations of amino acid residues within the pocket. How many? Between 1,000 and 1,000,000 different constellations of active site residues, again, without modeling the substrate. And we rank all these constellations by energy. So that means that we screen them for stability and pre-organization. And then we experimentally test only the 20 to 50 top designs with a variety of different substrates. Again, we're not targeting a specific substrate here. So this is a very simple algorithm. And yet for such a simple algorithm, the results were very striking and very surprising to us. What we saw, we tested 50 different designs. Here I'm only showing 4 different designs, again, relative to PTE, and their activity or fold change in activity relative to PTE and relative to these 4 highly toxic nerve agents. And what you can see, again, very, very clearly, this is in log scale, like the previous graph I showed. What you can see here very clearly is that these designs are much more effective than PTE. They reach as much as 4,000-fold improved activity relative to cycloserine or Soman in some cases, as you see here. So these were really unprecedented levels of improvement for a computational design. These designs were also broadly specific. What you can see here is a specificity profile of some of the computational designs relative to a bunch of substrates here. These are the 4 nerve agents shown here. What you can see is that these 4 designs exhibit very broad specificity against all of these nerve agents. And this is important for therapeutic purposes, of course, because typically in a nerve agent attack, we don't really know what substrate is there. We want the broad specificity countermeasure. And this is what these enzymes provide us. This work was done in collaboration with my friend and colleague, the late Professor Dan Tufik, who's an expert in vitro evolution. And he had worked on PTE for almost a decade before we collaborated on this project. And during this period, they were able to generate, using in vitro evolution processes, a variant that was therapeutically relevant only for one of these substrates, only for VX. By contrast, here, within 2 months, we reached therapeutic levels of activity for all of the substrates we tested. So obviously, FuncLib is much more effective in generating functional diversity and improved activity than in vitro evolution. But next we ask the question whether evolutionary processes were even capable of reaching some of the FuncLib solutions that we isolated in this process. The reason we ask this question is that if you look at the FuncLib designs in molecular detail, what you see is what you see here. So the wild type is shown in gray, and the mutations are shown in purple. This is just one mutant out of the set that we tested. And you can see that the mutations form a dense constellation, and that several of the mutations are small, histidine, in both of these cases, to large mutations, tryptophan and arginine. And these mutations are close by, meaning that we expected these mutations to have, may exhibit incompatibility between them, that they may not be compatible with one another. So here we turn to mutational analysis. The area of each circle is the specific activity, the relative activity of the enzyme that we tested in the lab. So the wild type enzyme is, as I mentioned, it exhibits quite low activity against the specific substrates we tested. Here we introduce each of the 4 mutations that I showed in the previous slide, each of them singly, as though this were an in vitro evolution process. So you can see that each of them improves the activity relative to the wild type, which would be very encouraging if this were an in vitro evolution process. And one of these, the histidine to tryptophan mutation, really improves it quite dramatically. Again, very encouraging for if this were an in vitro evolution process. So now if this were an evolutionary process, you'd ask the question, well, can I combine these mutations to form doubles? And this is when you reach this frustrating, what's known as the fitness valley in evolutionary trajectories. If you combine any of these, all the doubles are worse than the single point mutation histidine to tryptophan. And in fact, 3 of them are worse even than the wild type. So this would be incredibly frustrating, and it's often observed in vitro evolution campaigns. But what you can see is that if you combined this double mutant here with this double mutant here, you'd get the quadruple mutant, which is the one we designed using FuncLib, which is the best in the set. So this is known, such cases where combinations of mutations that individually decrease activity, when you combine them, improve activity. This is known as synepistasis. It's a very difficult form of interrelationship between mutations that is rarely observed in evolution, because it means that this typically reaches fitness values that cannot be crossed by evolutionary processes. So now it's clear that FuncLib cannot just generate improved variants compared to in vitro evolution. Some of the variants that FuncLib generates are simply unreachable by evolutionary processes. So I started this section by showing how evolution needs to increase, monotonously, one step at a time to reach a plateau. In effect, when you look at an active site, some of the mutations are much more rugged than that, and the paths may lead to fitness valleys, as you see here. But with FuncLib or design processes, we can reach the peak in one go without actually visiting any of these intermediates, which are suboptimal. So both PROS and FuncLib are enabled on web servers, and other methods from our lab are enabled as web servers, and we try to do that for any method that we find to be reliable and computationally tractable for our servers to handle. And what you can see is that over the years, literally thousands of academic users have used our methods, and this is very encouraging. We also see papers coming out where at the heart of these papers is the ability to use one-shot design processes using our servers in order to dramatically improve either the expression levels, as in this case, stability also in this case, of therapeutic enzymes, or their activity levels, as in this case. In some of these papers, I read these papers and I don't actually understand, these are systems I don't actually know beforehand. I get to learn about new systems, and it's very exciting for me to see how our methods can impact fields that I've never worked in before. So with that, I'd like to move to still unpublished results. And these results, this study, which has taken us several years to complete, and it's now under review, this study was motivated by our observation from PROS and FuncLib studies that when we apply PROS and FuncLib, in many cases we find that between 1/3 and 1/2 of the proteins that we test experimentally exhibit higher activity levels than the starting point wild-type protein. And that begged the question: if we designed, let's say, a vast library of proteins, let's say enzymes, how many active enzymes could we design, and how much structural diversity we could design in an enzyme? So here we targeted we didn't want to design just, let's say, a few dozen different enzymes. We asked the question, what would happen if we designed literally 1 million different enzyme structures? Obviously, there's no practical way of synthesizing so many full-length genes that encode for 1 million different enzymes. This is simply impossible using today's technology. So in order to synthesize so many genes, we turn to antibodies for inspiration, how to synthesize so many diverse genes. And why antibodies? So in response to an infection, our bodies are able to generate billions of unique antibodies to counter the molecular structures of viruses, bacteria, and other pathogens. So how do our bodies produce so many different and substantially different structures? They do this by the recombination of gene fragments. So what you can see here, these are known as V segments, D segments, and J segments. In order to generate a full-length antibody, you randomly select one of these red fragments, and you randomly combine it with a few green fragments, and then with a randomly chosen yellow fragment. And you do that for the heavy chain, and then you do something very similar for the light chain, and you recombine the light and heavy chains also randomly with one another. And if that produces well-behaved and active antibody against your target, that gets selected and then propagated in our immune system. This is an amazing system. It literally means that we can generate a huge diversity of different structures using very short genetic fragments and their recombination. But obviously, in order for that to work, these segments must be compatible with the red segments must be compatible with many of the green segments and so on. This means that all of these segments must be, to at least some extent, modular. So that's the guiding principle in how antibodies are able to, in how our immune systems are able to generate so many different antibodies. So we'd like to implement such a system to generate modular fragments of enzymes, but literally there's no enzyme family that is generated in nature in such a way. And actually enzymes are not known to be modular to the extent that we see in antibodies. So the key point that we ask in this study is, can we take an enzyme family and design the fragments of this enzyme family such that they become modular. So for instance, we can segment an enzyme, let's say this enzyme structure, into 4 pieces, yellow, red, blue, and green. Can we design different fragments for each of these pieces and then find the pieces that can be recombined, all of the yellows, all of the greens, blues, and reds, to generate a very large diversity of enzymes? How does this work? This is a method we call cadence. So we start by aligning all the structures of a given enzyme family. Here we worked on xylanases. These are enzymes that break down xylan, which is one of the primary cell wall polysaccharides in plants. And it's an enzyme that's interesting for biomass degradation and energy and biofuel production. So we align all the structures of xylanases we have access to, and we segment them into pieces as you see here. Each of these are different pieces from different xylanase structures. We then design each of these fragments using PROS to stabilize the fragment, and using FuncLib in order to diversify the active site aligning residues. Next, on the computer, we combine all of these fragments, all the yellow fragments with the green fragments, the red fragments, and the blue fragments, in order to generate a vast diversity of enzymes. Let's say tens of millions of different enzymes. And for each of these combinations, we compute the energy of that structure. And next we ask, what fragments generally give me low energy combinations or low energy full-length structures of enzymes? And we select those fragments for modular assembly. So we select those fragments that build about 1 million. Out of these tens of millions of different structures, we select those fragments that would build the 1 million lowest energy full-length enzymes. I'll come back to this important point of how we select these fragments in just one slide. But for the time being, as I walk you through the cadence pipeline, this is what we do. We select those fragments that would build 1 million low energy structures. Then we order these fragments. You see here are all the yellows, here are all the greens, et cetera, and we combine them in a test tube to generate full-length enzymes. So each of the molecules in the test tube encodes for a single full-length enzyme. Now we encode this library in yeast on the surface of yeast cells, and we use what's known as an activity-based probe, which was generated by Herman Overcleft from Leiden University. This activity-based probe is based on xylan. The molecular structure you see here is a xylan disaccharide, as you see here. And what's unique about these activity-based probes is that when you incubate these activity-based probes with active xylanases, they actually covalently attach to these active xylanases and label them with a fluorescent molecule, as you see here, fluorescent dye. And that enables us to use very straightforward fluorescent activated cell sorting in order to sort the active xylanases from the inactive ones. So now we have in one test tube all the active xylanases within yeast cells. We sequence all of them using deep sequencing techniques. And then we look at all these sequences, and all the sequences that were not found in this screen are presumably inactive, and we try to find discriminators that discriminate between the active and the inactive population. Keep in mind that in this study, we're not interested in finding, let's say, the world's best xylanase. What we're interested in learning are effective rules for enzyme design. So we'd like to know why those enzymes that were active, whereas the remainder were not active. How do we test that we actually learned useful rules? We apply this activity predictor in order to design a second repertoire and repeat this process in order to see whether our yield of active enzymes has improved. So before I show you the results of this study, let's go back to this question of how do we select fragments that are compatible with one another. So when we combine fragments of all sorts of shapes and sizes, as you see here we're combining 2 blue fragments. This blue fragment is identical to this, but it's different from this. So you see the blue fragments here are here in columns, whereas the red fragments are here in rows. And this is essentially a 2x2. We're combining 2 blue fragments with 2 red fragments. So when we combine a variety of different fragments of all sorts of shapes and sizes, some of them are good, some of them are bad in terms of the energy and compatibility, but some of them can be literally terrible. As you can see here, the red and the blue fragment are intertwined, meaning that there is a huge steric clash between them. This would be a completely impossible combination, leading to probably unfolded and unstable and definitely inactive enzyme. So we'd like to eliminate as much as possible any combinations that are bad in terms of their energies. How do we do that? So I mentioned that initially we combine all of the fragments, all of the design fragments to generate full-length enzymes on the computer and rank them by energy. Then we label the top energy, the best energy, 10% as being good, whereas the worst enzymes as being bad. And then we train a very simple machine learning neural network in order to predict, based on the fragments we use, in order to predict the outcome of the energy, the Rosetta energy. Is this combination stable? And is the active site also stable? And then after we've trained this very simple neural net, we ask it to rank each of the fragments individually. We ask, is this blue fragment compatible with as many other red and green and other fragments as possible? If it is, great, we keep it. If not, we throw it away. And we rank each of these fragments and then combine them. We use that in order to generate the experimental repertoire that I mentioned on the previous slide. How do we know that this works? Of course, we don't want to go to experiment before we have a sense that this neural net approach actually selects compatible fragments. In order to do that, we can compute the energies of the neural net selected fragments. And you can see that the energies, the atomistic energies computed by Rosetta are much lower and much shifted to the left, much better than random fragments combined before the neural net selection approach. So this gives us confidence that the neural net actually does the right thing of selecting fragments that are compatible with one another. So does this work experimentally? So Rosalie, who led this project, went to the lab and tested whether this strategy works. And after deep sequencing, she found more than 3,000 active designs in her set. And these active designs were incredibly diverse. They used more than 700 unique backbones. When she tested the enzyme's identity to any natural protein in the sequence databases, she found that many of them exhibited, at most 50% identity to the nearest natural enzyme, which means that they comprised as many as 160 mutations from any natural enzyme. So they were incredibly diverse, very far from natural enzymes. They also source fragments. So we're combining 4 different fragments. Most of them actually source fragments from 4 different natural enzymes. So again, using very complex combinations. So you could also ask whether these, if all these mutations were outside of the active site pocket, this may not be very interesting for functional diversity. But when we look at the structures of these designed enzymes, which are selected because they're active, what we see is that the active site pocket varies dramatically between the designs. Here is a model of the substrate bound within the active site pocket. And you can see that each of these 4, randomly or arbitrarily selected enzymes exhibits different shape and electrostatic properties within the active site pocket. So these enzymes are not just surface mutations relative to one another. They actually have substantial changes in the active site pocket, which may yield new substrate specificities. So this is the key point about this study. So as I mentioned earlier, we're not interested in designing the world's best xylanase, where we want to understand what makes some of the designs work, these 3,000 designs work, whereas the majority of the other designs are not working. They're failed designs. So Rosalie used a variety of different energy and structure characteristics, structure features, in order to try to discriminate between the active population, shown here in blue, and the inactive population, shown in red. What you can see is that these are just 4 of the almost 100 features she tested. What you can see is that none of these features on its own gives you a very good discrimination between active and inactive. There's some discrimination, but none of them is very good. But then if you combine 10 of these features together using a standard logistic regression model, what you see is a very clear separation between active and inactive designs. And this is great. Now we have what we call an activity predictor. Given a new structure, a new design structure, we can determine whether we predict it to be active or inactive. So using this new activity predictor, Rosalie went on to design a new repertoire. Now she designed only 300,000 structures. In the first repertoire, she designed 1 million. Now she designed only 300,000, and she tested them experimentally. And she did see a huge increase, about an order of magnitude increase in the number of, or in the yield of active designs. She first tested with xylobios, the same xylem-based activity-based probe that I mentioned earlier, and again, saw much improved activity. And then she used another probe called celobios. And what you can see here is that she detected nearly 10,000 xylanases in her repertoire, and nearly or more than 3,000 new cellulases in her repertoire, even though the original family from which she designed this enzyme only exhibits very low cellulase activities. In her designed repertoire, she found more than 3,000 unique cellulase designs. They're again very, very diverse relative to one another and relative to naturally occurring xylanases, as you see here. And the bottom line here is that we got from this second repertoire more than 12,000 active enzymes, an order of magnitude improvement in success rate. And if you combine both repertoires that Rosalie designed, she got more than 16,000 unique and active enzymes. Keep in mind that just a few years ago, designing such a complicated and sophisticated enzyme, a single one, was considered to be a major feat. Now we can design more than 10,000 in one go. I also mentioned to Rosalie once that if she computes her average rate of active designs in her PhD is about 1 active enzyme per half hour of her PhD. So that's not too bad in terms of productivity. Next, we wanted to do an independent test this independently of this repertoire, this time not designing a huge repertoire, but designing a small set, let's say of just, in this case, 27 unique xylanase designs. This time she's not designing them through modular parts, she's just designing each enzyme to be as good as she can. Out of these 27 designs, she found that 25 were active, which is an amazing success rate for such a complicated design challenge. And many of them were as active as natural xylanases from thermophiles. Here she tested these enzymes relative to the native substrate. This is really xylan taken from wheat and beechwood. And you can see that her designed enzymes are essentially just as good as xylanases taken from natural thermophilic organisms, which are literally the best xylanases we can come up with. And again, these designs have more than 100 and as many as 150 mutations relative to any natural enzyme. They're very stable, very highly produced. These are excellent enzymes, even though they were completely computationally and automatically designed. Yes, so this is the point. And one of the visions for what are we going to do with this strategy? My hope is that in the future, when people want to find, when researchers want to find a new activity or to change the substrate specificity profile of an enzyme, they would not just look in nature to find this new activity. They would use a strategy such as this, which would enable them to generate much more active and also much more stable enzymes, again, in one shot. So we were very encouraged by this ability to design various structures in an enzyme family. And in parallel to that, we asked the question, can we use a similar strategy in order to design just a sequence, just to introduce sequence variants within the active site pocket of a protein? And here, again, the underlying question is, how malleable are protein active sites? Or in other words, if I designed this sort of diversity, the diversity I showed you earlier, but only using sequence changes, not structural changes within an active site structure, how many functional variants would I be able to pick out from such a library? And here, let me just get a drink for a second. Excuse me. Our model system for this question was diiP. diiP is really a model system for mutational tolerance in many different studies, as this one, for instance. The chromophore in GFP is buried within the core of the beta barrel, as you see here. And the reason it's such a good model system is that the readout is optical and very straightforward. You shine light at a specific wavelength, and the protein is green. It emits green light, as you see here. So most of these studies, most of these mutational analysis of GFP generated random mutations across the protein. And most of the tolerated mutations are obviously on the surface of the barrel facing outside into the solvent. And such mutations are unlikely to impact the spectral properties of the protein. They're not likely to impact its functional properties. And of course, people have not really detected functional changes in these libraries. What we wanted to do in contrast was to only target the core of the GFP protein, really introduce mutations that either touched chromophore or second shell mutations that touch positions that touch the chromophore. So here we're focused on 14 mutations that face the chromophore. This is the most sensitive part of the protein, of course. And we wanted to design a space of about 5 million different designs. And again, using the same atomistic and machine learning strategy that Rosalie developed, we tested whether the designs, at least computationally, whether they're likely to be folded and active. So here, this is the distribution of energies if we just introduce random mutations within the core of the protein. And this is the wild type energy. So the vast majority of random mutations are much worse than the wild type, whereas the design mutations are much, much better. The combinations of design mutations are much better than the wild type. So this was, again, encouraging for us that this designed library would be folded and potentially active. The design library is quite complex. You can see here the mutations. Some of them are not radical, valine to isoleucine, leucine to valine, but in some positions, we introduce very radical mutations from tyrosine all the way to alanine, or from threonine to glutamate, et cetera, et cetera. So it's a very complex library. We encoded this library and then tested it experimentally, and this just shows, the bottom lines of this library. We're looking for mutually compatible low energy combinations of mutations, and also for mutations that are compatible with the chromophore, and we focus diversity only on the active site. This shows the experimental pipeline. Very, very briefly, we generate the library, then express it in standard bacterial cells. Then we use FACS sorting because GFP is green. We can use cell sorting in order to isolate the active population, and then Illumina deep sequencing to find the active population. So again, to our surprise, when we selected the active population, we found 16,000 unique designs that were active. And this is literally 2 orders of magnitude more active designs within the active site pocket of GFP than are available in public databases of fluorescent proteins. So this is, again, one-shot design and experimental screening. You can see here that even at 6 mutations from the wild type, about 15% of the GFP designs are active. They're fluorescent proteins. 6 mutations away from the wild type within the active site pocket is a big number of mutations. We wanted to have a look at the sequence landscape that's spanned by these design mutations and compare it to naturally occurring fluorescent proteins, as you see here in gray, and to a previous set of designs generated using AI-based methods by George Church. And you see that those span sort of subspace of the wild type population of active site mutations in the fluorescent protein database. By contrast, our designs span a very large space of mutations. Of course, we're not covering all of the wild type diversity, but we're covering a huge space of other mutations that are probably not covered by, have not been covered by evolution. So next, we wanted to know whether these mutants also present or exhibit different spectral properties than GFP. So we selected 70 different designs and tested them experimentally. We purified each one of them, and one of the fun things about GFP is that you can literally see the protein when you express it in bacteria. You can literally see that it's fluorescent. And then we tested their spectral properties, starting with quantum yield or brightness. What you can see here is that enhanced GFP, the variant we started with, exhibits this level of quantum yield, shown here in dashed lines. And 28 of the 70 designs we tested exhibit higher quantum yield or brightness than enhanced GFP. So they're clearly brighter and potentially more useful than enhanced GFP. We also saw very large changes in excitation spectra. We selected all of the designs because they emit light in the green wavelength, so they all emit light in the same wavelength. But you can see that the excitation spectra are vastly different from enhanced GFP and from one another. So we see huge changes in excitation spectra. And then we measured a bunch of other characteristics. Here is functional thermostability. Some of them are boiling resistant, fluorescence lifetime, photostability. In every physical measurement that we tested these designs, we saw wide and potentially quite useful diversity in this library. So keep in mind, this is one designed library, no iterations, and it encodes huge functional diversity. So I'll just summarize the key take-home points from my talk. What I showed you in the first half of my talk is essentially what I believe to be a very general optimization strategy. You can apply computational design within the active site pocket to improve activity using FuncLib, web accessible again, and outside the active site pocket to improve stability, PROS. And that's great. Most of these designs are improved activity relative to the parental protein, including in cases where in vitro evolution, this is critical, where in vitro evolution failed. They actually improve our ability to design proteins. And usually you don't actually need domain-specific knowledge in order to use these methods. Simply a structure and some understanding of where the active site pocket is in your protein. What I showed you in the second part of my talk is something that I call, these are new methods. We haven't tested them extensively, but I believe that these will be really a game changer for what I call multi-scale. This was not available before, but multi-scale protein design. We can much more efficiently explore sequence, structure, and function space than existing methods. So around every natural protein, we can now design literally millions, potentially even billions of different sequences and structures that exhibit different activity profiles. And now we're testing this with antibodies, nanobodies, a variety of different enzymes. I think this is a very exciting time for computational design and synthetic biology. And with that, I'll close. Thank my many collaborators. So these are just some of my collaborators who were involved in the studies I showed you, and my team members, and you for your attention.
Thank you so much for speaking to us today. I would like to know if there are any questions. Sebastian, do you wish to ask something?
Yeah, I have a number of questions. The optimization is something very important. For those people who are working on recombinant protein production, for example, in E. coli, there are some very trivial aspects. And one of them is the codon usage.
Yes.
That may be quite different between the original organism and E. coli. And just by adapting the codon usage to E. coli usage, we may increase a lot the production of recombinant protein. So in your approach, this is completely ignored, or it is as a background that you always express optimized synthetic genes or whatever?
Yes, absolutely. So what I showed you were cases in which the wild type protein, not the wild type gene, was codon optimized for E. coli, and the expression levels were, as I showed you, they were terrible. And the designs were each individually also optimized using the same strategy. I think we use emboss typically for codon use in E. coli. When we use yeast, we codon optimize for yeast. So these are cases in which codon optimization on its own was clearly insufficient.
Yeah, okay. Yeah, that makes sense, of course.
Yes.
Related to your approach. I have another question. In classical enzymology, it is usually said, and we say that to our students, that the Km of an enzyme for its substrate is usually in the order of magnitude of the concentration of the substrate within the cells. I think that if we browse databases such as Brenda, for example, we find most of the examples fit relatively well to this empirical rule. So do you take this into account in your optimization process, or this is completely independent on?
It's a really good question. We are not taking it into account because we're not really modeling. Typically, we can model the substrate. We typically don't do that. We don't model the substrate within the active site pocket when we optimize using FuncLib. What we observe, so I mentioned that, okay, so what we observe in many cases is that we get a diversity of KMs and KCATs where the catalytic efficiency, the KCAT over KM is sometimes maintained. So, and that's quite interesting because you're right that the cellular levels of substrates are often maintained homeostatically. But I mentioned these lignin processing enzymes. In such cases, the substrate is extracellular and its levels may fluctuate wildly. Also in industrial processes, you can imagine that you start with a high level of substrate, but with time it's depleted to very low levels. And in such cases, you may want two enzymes, one that has low Km and high Kcat in order to break down the substrate when the concentrations are low, and the other which has high Km and low Kcat, right? So that's what we sometimes observe, that we sort of maintain catalytic efficiency, but really play around with Km and Kcat values. And this may be very useful actually in vitro, I would say, processes. I'm not so sure, but you're right. I'm not so sure this would be useful for within the cell.
Yeah. May I have, yeah, I have another question.
Sure.
If it is possible. You showed a structure. I think it was an alpha-fold structure with a heme and ions. That heme was docked as a ligand to the protein, I guess.
Yes.
And so your conclusion is that, in fact, it is a non-covalently bound heme. It's a heme B, I think.
No, it's actually covalently bound. This was just.
It is covalently? Okay.
Yes.
So but my question is that this type of ligand, in fact, does not really contribute to the overall folding of the alpha chain, or does it contribute at some step?
I would say I can't address this question experimentally, but what I would say is that the enzyme has a huge pocket without the heme, right?
Yeah. And it's close when or.
So we don't, but the model for that structure, when you generate a heme or this enzyme using AlphaFold, what you observe is a huge cavity within the active site pocket. So that may be worrying because do you actually know that this would fit with heme? But then when you superimpose heme within the pocket, what you find is that all the rotamers are literally in position to hold the metal ion, the iron ion in the middle, and the heme cofactor. So it's literally as if AlphaFold knows, it does know, that this is going to hold heme in the middle, and the entire structure is essentially waiting for the cofactor. The same is true in metal binding sites. You see these constellations of histidines around the metal ion, which is not there and not modeled using AlphaFold.
Yeah. Maybe one more question.
Sure.
In your approach of assembling modules of protein to try to optimize and generate hundreds of thousands of structures, it seems to me that the output, at least in the examples you show, tend to eliminate the loops or the main loops of in the structures. Is it just something that I missed from your structures, or?
They certainly include, actually, you know, the perspective from my lab is that the loops are the most important part of an enzyme.
Okay, I fully agree.
Yeah, okay.
Yeah, absolutely, fully agree. Of course, when the loops are overlapping or something like that, there is some problems.
Exactly.
But, because there is a key related problem or question is that most of the time these loops are important for protein-protein interaction.
Absolutely.
And, is it something you are going to address?
Yes.
Yes.
Yes, we are working on antibodies. We have
antibody, yes, but antibodies something maybe slightly different.
yes, but we do work with enzymes. You know, our main efforts are actually concentrated on the loop regions because those are the most difficult to model, and also because they're the most important for specificity. We also have worked on designing new specificities in binding proteins using the loop regions.
Yeah.
And we generated specificities that were at least 100,000-fold relative to the starting point by designing a very rigid loops in the binding interfaces of, do you know the collagen endonuclease immunity? Yeah.
Well, congratulations. It's absolutely beautiful work.
Thank you very much.
Thank you so much.
I have a question as well.
Yes.
So my background is not in biology, so this seems very exciting to me. So I guess in computational design, what you're aiming for is for a high yield. So you want to have active structures. And you did spend some time talking about how to ensure that you get active structures. My understanding was that this was a post-processing step. So you first design some structures, and then you have another set of automated tools that check them and select which ones are active. So this seems to be a little bit of a waste of effort. So I was wondering if it is possible to actually change the computational design process itself to produce active structures right from the start. And even more so, whether we can actually direct the design towards the direction that we want. So not just active structures, but for example, active structures that in addition are very resilient to heat or whatever else feature we want. Thanks.
So this is an excellent question. I just want to clarify though that in the first part of my talk on PROS stability and then FuncLib improvement of activity, these designs were designed for in the case of PROS for stability. They were not. There was not this process of first let's generate diversity and then computationally select the best ones. It was literally I'm designing now for native state energy, low native state energy. So in that case, it was fine, and it's a quite efficient process. There's no, I don't think this is inefficient specifically. With FuncLib, you're right that we're not directing to a specific substrate. So you're right that if we could direct to a specific substrate, we could make FuncLib more efficient. The problem with that, and this was something that I was worried about for many, many years before starting on this FuncLib, that in enzyme design, we often don't have a good chemical understanding of what is known in the field as the transition state. So we don't just need to know how the substrate docks within the pocket of the enzyme. We actually need to know what is the most unstable structure in the process in which the substrate, let's say, breaks. Let's imagine this substrate needs to break. We need to know what is the structure of the substrate when it breaks relative to the enzyme active site. And in most cases, we just don't have that information. FuncLib is a sort of a practical tool saying, yeah, we're likely not to have this information for most of the enzymes we care about. What can we still do? So it doesn't direct to a specific substrate. And you're right, this is a major limitation. We're trying to overcome it now. We have a sort of a geometric learning project where we're trying to address this. We can talk about this offline if you're interested. We don't have a solution yet. In the second part of my talk, sorry for the lengthy answer. In the second part of my talk, I mentioned that we're building the fragments and then enumerating them. And then, as you said, we're selecting them posteriori for stability and for activity computationally before we go to the lab and test them. And here too, we were thinking about this problem for many months of how do you design a fragment of a protein such that it would be compatible with as many other fragments as possible. And that's not, I don't know how to frame this as an objective function for design, right? This is something that I only know how to address sort of retrospectively. Having designed fragments and recombined them, I can now sort of look at them and say, okay, so this fragment usually combines with other fragments to produce a low energy structure, I'll take that in. You're right that if there were a direct way to design the fragments such that they would be compatible, you know, all of them such that they would be compatible with all of them, that would be beautiful. But I simply don't know how to do that. If you have ideas for that, I'd be super interested.
No, I don't really have any ready answers. I was just wondering if this makes sense in your case.
Your question was, you know, it went to the heart of the matter. You're absolutely right. This is the major problem we try to address, is our sort of inability to frame this as a you know, mathematical objective function for optimization, and sort of only in this roundabout way could we do this. You're absolutely right.
Okay, thanks.
If that's all, then we will conclude the lecture, unless anyone else has something to add or a question to ask. Okay, well, thank you so much for joining us today, and this was very, very interesting. And thank you so much for making yourself available and talking to us about all of this. It's very interesting, definitely.
Thank you.
Thank you. Thank
Many
you.
thanks, Sarel.
Good night.
Bye-bye.
All right.